iPhone has Wi-Fi and a touch screen and computing and a battery and a camera and an LCD display and uses the internet and World Wide Web. But where did all those things come from? And it's all the material from each one of those points that points to the science that was about. So a little bit below the picture of the iPhone is this collection of all these discoveries. But this is really at the top of the chart. The chart eventually gets down at the very bottom to discoveries more than 2,500 years ago of, for example, using 0 and 1, the first use of binary, the first lens. All of these things are absolutely needed, and everything that was done subsequently built upon these discoveries. So my contention, as I think it was with Flexner, is that the basic understanding about how the world works is absolutely essential for building on any other application or particular achievement. And I want to show you my favorite video. This was made, it's a little bit outdated now, actually, but it was made in 2014 by a group of students and postdocs at the University of California in San Francisco in response to a contest. This was a contest from FASA, which is the Federation of American Societies of Experimental Biology. And the contest was make a short video that shows the importance of basic science. And this is the winning group. You can see their names at the bottom. And as you listen to it, I think I hope you'll agree that it really is a rather remarkable little film, but it also gets this same idea across. So let's watch this short video.
It's a sunny day in San Francisco. We set up a table by the water and asked people this question. If it's the year 1960 and you had $10 to give to science, would you spend it on A, developing an affordable treatment for diabetes, or B, figuring out how bacteria protect themselves? The majority of people we surveyed wanted to spend their money on a diabetes treatment, but what really happened 50 years ago? The government decided to spend money on figuring out how bacteria protect themselves against viruses. The National Science Foundation, the NSF, and the National Institutes of Health, NIH, are two government agencies that allocate tax dollars to scientists. And they decided to fund groups of researchers who thought that studying how microbes protect themselves was an interesting question. At the time, there was no way of knowing how this basic research could affect human health. They were simply curious about understanding something about bacteria biology. So what did the scientists find? Normally, when a virus enters a bacteria, it adds its own DNA to that of the bacteria. In doing so, the virus takes over the bacteria and uses it to make more viruses. Scientists found that bacteria protect themselves from viruses by selectively cutting the viral DNA. This basic finding led to a scientific revolution. Scientists learned how to cut specific chunks of DNA, and they could use this technique to move genes around. They could even move human DNA into bacteria. And because bacteria are very good at making more of themselves, scientists can use them to make large quantities of human proteins from the human gene that they introduced. So why was this technique useful for modern medicine? Well, one breakthrough was the efficient production of human insulin by introducing the human insulin gene into bacteria. This was a very significant advancement in diabetes treatment. Before using bacteria to produce large quantities of human insulin, insulin was expensive, unsafe, and less effective because it was harvested from fetal cows. In fact, the first person to get an injection of cow insulin suffered from a severe allergic reaction. Because of a long line of federally funded basic research, human insulin is now an affordable treatment for diabetes. By asking how bacteria protect themselves, the scientists discovered that bacteria can cut DNA. This discovery was developed into a basic technique used in thousands of labs around the world to create new knowledge and to ask new questions that have led to all kinds of therapies and technologies, including insulin, DNA fingerprinting and biofuels, the human genome, growth hormones, cancer drugs, stroke medication, vaccines, HIV meds, antibiotics, and many, many more. Given enough time and funding, who knows what is to come? Many other health benefits and current discoveries would have been nearly impossible without federal funding for all kinds of basic research. Basic research is an investment in our future. It broadens our understanding of the world we live in, and the potential benefits are limitless. Unfortunately, science funding in the US has plateaued, decreasing purchasing power and significantly hurting our competitiveness and innovation. You may not know it, but you and your tax dollars are part of the discovery process. Thanks to you, scientists discovered that bacteria can cut DNA. And thanks to you, insulin, other drugs and diagnostic tools have been developed and are helping patients.
So thank you for supporting basic research.
We are the scientists who depend on you. And we are excited for the future that lies ahead.
Great. So, how does one make these discoveries? How does one enlarge our knowledge, our basic understanding of the world around us? Well, the Nobel laureate and physicist Enrico Fermi had a very interesting point of view on all of this. He said, if the result of your experiment confirms the hypothesis, then you've just made a measurement. If the result, however, is contrary to your hypothesis, then you've made a discovery. So the idea is that hypotheses are good, but there's a lot more to discover. And so one of the questions I'm going to try to address in the remaining part of the talk is, how do we make discoveries? And I think there's really two parts to these things. I think it's seeing something that you did not expect and realizing that it was an important thing to follow up on. So we never know when we're going to make one of these discoveries. So the only approach to that is to do lots of things. And so if you do lots of things, hopefully something interesting will come up. And I want to give you a couple of examples from Nobel laureates of this. So the first example I want to give are these two people, Robert Wilson and Arno Penzias. They were both working at Bell Laboratories in New Jersey in the United States. And Robert Wilson has described this many times, that he had a failed PhD project. He never really got answers, and he was interested in measuring microwaves in space. And when he got to the Bell Laboratory, he discovered that some previous scientists there had arranged to have built a 30-foot microwave telescope. So he decided to revisit the work he had tried to do in his PhD work. And when he and Arno Penzias started the experiments, the first thing any self-respecting scientist would do is make sure the equipment is working properly. So they realized that the Milky Way would be a great source of microwaves. And sure enough, they point the telescope at the Milky Way at night, and they'd get a wonderful signal. But now they wanted to see what zero was. They had a nice strong signal. Now they wanted no signal. So they pointed it at empty space. And to their surprise, they never could get to zero. There was always something there. And they He had a lot of controls. They thought they couldn't even start their experiments because their controls didn't work. Their calibration of their machine didn't work. And then they realized that the reason they could never find zero is because out in the universe, it's not zero. There is a background radiation. And this realization was really led to having the best information or the best background to understand the Big Bang, that there was a residue of the Big Bang, and it was this background radiation in the cosmos. And that's what they won the Nobel Prize for. Another person is John O'Keefe, more recent winner of the Nobel Prize, this time in medicine or physiology. And he was known for discovering cells that respond when an organism is in a particular place. These are called place cells or place neurons. And what in his Nobel Prize acceptance speech, he says that he was doing experiments when one day by mistake, he put the electrode in the wrong place in the brain. And when it went to this incorrect place, that's when he discovered these cells. And of course, he noticed it enough to say, well, maybe I should be studying these. And that led to the understanding of these particular cells that respond to an animal's position in space and led to his Nobel. The third example I want to give is a much more closer to my heart, and that is of one of the people that shared the prize with me in 2008, and that is Osamu Shimomura. Osamu Shimomura was an amazing man. He has written a book called Luminous Pursuit, which is a memoir, which I strongly suggest people go and look at because his life has been rather remarkable. As a 16-year-old in high school, he's told you have to quit school. That's it. You have to quit and work in a factory. And he does that. He leaves high school, he starts to work in a factory. And that turns out to be very good for him because the year is 1945. And he's living in the town of the city of Nagasaki, Japan. And by working in the factory, he is saved when the atomic bomb destroys the city. He describes walking home 10 miles from the factory to get to his home, by which time his white shirt has turned a very dark gray because of all the radioactive ash that has fallen on him as he has walked home. Talks about his grandmother making him take a bath, and he credits her with probably saving his life by getting rid of all that ash and radioactivity. But after World War II, there was no place for him to go to college because everything was destroyed. Eventually, they were able to build the pharmacy school, which he went to. He said he wasn't interested in pharmacy, but he wanted to go to college. Afterwards, he goes and he works as a technician in a laboratory in Nagoya, Japan. And it's there that he really finds his life work, because one of the projects he's given is to understand how a small crustacean called Cypridyna, which is bioluminescent, that is, it generates light, how it does it. And he's very fortunate. An experiment he's doing works out appropriately, and he's able to find the critical compound that allows this crustacean to generate light. And so he goes off for the whole rest of his life and studies how different organisms biochemically can generate light. It's a fascinating problem. Lots of different organisms, fireflies, glowworms, jellyfish, crustaceans, fish, fungi, bacteria, can generate light, and they all do it differently. So if you understand one, you really have to start all over again to understand another. And that's what he studied his entire life, how organisms can generate light. And one of those organisms was this jellyfish, Aquaria victoria. And he set off in 1962 to try to understand how it was that this jellyfish produced this beautiful green light. And when he did, he was a very good biochemist, but every experiment he did failed. No matter what he added to the preps, he could not isolate the compounds that were needed to generate light. After a whole summer of frustration, he does the experiment one more time. It has failed once again. And he decides it's late at night, he's hungry, he wants to go home for dinner. And so he goes, he cleans up after himself. He throws the samples in the sink, which had some seawater, probably other things in it as well. And he turns off the light and he's just about to leave when he looks at the sink, and to his surprise, everything is glowing. He thinks about this for a while and realizes there must have been something in the sink that he never put in his preparation, and that's the missing component that was needed to generate the light. He wonders what's in seawater that I haven't used, and he realizes that it is calcium. And so he does the experiment again a couple days later, and every time to his sample, he squirts in a solution with calcium ions, he gets a spark of light. And he can use this to purify the protein. And the purified protein, he names after the jellyfish, he calls it aquarin. And aquarin plus calcium produces light. So a completely accidental discovery allows him to sort this out. But he's not done with his accidents, because the problem here is that the light that's produced is the wrong color. The jellyfish are green, but this light that's produced by this reaction is blue. He thinks about this for a while, adds calcium to all his other samples, never gets any green light out of them. And then he thinks maybe there's a molecule that converts the blue light to green. And so he takes a handheld lamp and he looks at all of the samples, shining blue light on them. And he finds that there's one of the fractions, one of the samples, does in fact give green light when stimulated. Doesn't need calcium, doesn't need aquarin, it just needs blue light and you get green. So two wonderful accidental discoveries. He writes all of this up in a paper in 1962. And in the paper, most of the paper is about purifying aquarin, but he has a footnote. And the footnote says, there's this other protein that is stimulated by blue light, and you get green light out. And I'm going to call it the green protein. Well, we don't call it the green protein anymore. We call it the green fluorescent protein, because fluorescence is taking color, one color, blue, and changing it to green. And this abbreviated as GFP. And so what he was looking at in the jellyfish is the reason the jellyfish was green, was giving off green light, is because the blue light never got made. The energy from aquarin plus calcium went directly to GFP, was converted to green light, and so the jellyfish produced a green light. A wonderful discovery that basically sat around for many, many years. 1962, no one was doing any cloning, no one knew how to isolate these things. And I was one of the many, many people that had never heard of this before. And then I heard a seminar in 1989. And I heard about, in this seminar, this story that I've just told you about Osamu Shimomura and the discovery of GFP. And I had been doing genetic work on a small, transparent worm called Cinerabditis elegans. And I realized that because the animal I worked on was transparent, and because I wanted to know where genes were turned on and off, all I had to do is have those genes, hopefully, make GFP. And then I simply would shine blue light onto the molecule, and wherever it was, I would see it because that's where I would see the green. In other words, this would become a lantern. And I got very excited about the seminar, about this result. I didn't get that excited about the seminar because I stopped thinking about or listening to the seminar, and I just imagined all the experiments we could possibly do. Got back to my laboratory and found out that Douglas Prasher was in the process of isolating the DNA for GFP. And so we decided to collaborate. But it wasn't until 3 years later that a new graduate student, Ia Iskirkin, came into my lab who had had some experience with fluorescence that we were able to finally do the experiment. And so her first experiment as a new graduate student was to try to put GFP into E. coli, into bacteria. And it worked. And she was able to get bacteria, this is one of her pictures from that very first day, that were fluorescing whenever we shone blue light on them. But we were very excited because what this said is, you don't need anything except GFP. All you need is GFP and blue light. A little bit later, we put it into worms. And now for the first time in a living animal, we could actually watch the cells that we were studying. And this also meant that we could take these living animals, mutate them, and look for changes. More cells, fewer cells, cells in the wrong place, cells that grew more processes than they were supposed to. There was so many opportunities once we were able to see the cells that we were excited about. And GFP has been put into Drosophila and zebrafish and mice and many, many other organisms, untold numbers. And because GFP is a small molecule, it can go everywhere in the cell, and it gives you some spectacular pictures of cells like this Purkinje cell from a mouse brain. Now, the really wonderful thing about GFP as a marker for cells was the fact that once you put it in an organism, you could see it in that organism because shining blue light doesn't hurt them. You didn't have to treat them in any way. All you had to do is shine light on them. And it also meant that they would pass this along to their progeny. And this opened up lots and lots of opportunities. And I want to show you 2 short movies to give you an idea about what it is to actually watch these, use GFP to watch something happen in living organisms. Both of these movies were put together by a graduate student who I've never met from Canada named Rosalind Silverman Gavrilla, and she nicely put these films on the archive of the American Society for Cell Biology, and that's where I was able to get them. Now, the film on the left-hand side of the screen is a picture of the nuclei dividing in the early fruit fly embryo. And GFP has been attached to one of the proteins that makes up the spindle, the apparatus that separates the chromosomes. So when I start the movie, you're going to see 4 rounds of nuclear division, and you'll see the spindles form and then come apart after the division is complete. So the spindles are forming, and then it's over, and it's very much sped up so we can see all 4 rounds here. And she actually names this movie In Synchrony. because that's one of the most striking aspects about what's happening here. And being able to see this live is that it's all happening together. And so it immediately, as this loops around again, it immediately invites the question of how is this synchrony coordinated? And of course, people now know quite a lot about the molecular controls of cell division, nuclear division in this case. Now, the movie on the right is falsely colored, which means that if there's more GFP, you're closer to the red end of the rainbow than the blue end. So blue is a little bit of GFP, green is a little bit more, orange a little bit more, yellow a little bit more, orange a little bit more than that, and the most is red. So it's falsely colored. It's a convention to look at different levels of GFP, but it's still GFP that was used. And this is a movie she names after the Van Gogh painting Starry Night, and I think you'll see it. Here, GFP is attached to a small peptide called a nuclear localization signal. And what this does is whenever there is a nucleus, as in the beginning image, the GFP is sucked right into the nucleus. So all you see are the nuclei within this embryo of fruit flies. But when the nuclei start to divide, the nucleus is going to open up, going to dissolve away, and the GFP is going to go everywhere in the embryo and then reform and be sucked back into the nucleus when the nucleus reforms. So let's watch that. So now everything's going away. Now it's everywhere in the embryo. The nuclei are going to reform and start taking up the GFP. And then they break down again. And here it's not synchronous. It's interesting. It actually goes from the lower right-hand corner to the upper left-hand corner. And what's happening here, we believe, is that there's some slight damage here. So that's not synchronous. It really should be more synchronous here. But nonetheless, it's a very pretty picture. And it shows how sensitive we can use GFP to pick out various aspects here. Now, GFP has been used tens of thousands of times, many, many papers, many, many studies. Some of the work has been fundamental. And to give you an example of one of my favorite aspects of fundamental, Tony Hyman and Cliff Grangwin in 2009, the year after the Nobel, using that same worm that we had studied, they were looking in the early embryos, and they were interested in a protein that was found in the cytoplasm of the zygote, the fertilized egg. And what they found, to their surprise, is that it acted, even though it was soluble in aqueous solution, it acted like it was separated from all the rest of the cytoplasm. No membrane, but just a separate thing. They referred to this as a phase-separated particle. And here's some of the pictures. It looks as if it's oil in water, but it's not. It's two different water-based parts. And what this meant is they discovered a new organization of the cell cytoplasm. And people are now really starting to understand what all this means and what the implications of this separation of components means, not only for the cytoplasm, but also for the nucleus, because these are also found in the nucleus. There's also questions that are not as fundamental as more applied. So one that I like quite a lot that also came out in 2009 was a study of how the AIDS virus, HIV, gets transferred from one cell to another. Now, this is clearly a very important thing to understand, because if you understand how a virus transmits from one cell to another, you might be able to interfere with it. Now, when we usually teach introductory biology, we say, oh, viruses infect cells and the cells explode. Well, if the cells explode and the virus is all over the place, then an antibody might be able to get at that. But what really happens here? Well, in this example, the AIDS virus infecting this mouse cell has GFP in it. Now you see the beginnings of a viral particle. It's going to be transferred to another cell. No explosion, no general distribution. So a little bit harder to control, but maybe some insights into how this transfer takes place is going to be important in trying to control how the virus is transmitted. So both of these things, fundamental research, applied research, come out of studies that started with the basic useless information of this jellyfish protein. And we can say fundamental research gives applied, but it's really a two-way street because the applications then help the fundamental research and it really works back and forth. But there's actually a third possibility. And that is things you never imagined, or at least I never imagined, the unexpected. And I'll give you some examples of some of the unexpected things. People in Japan decided that they were going to put fluorescent protein into silkworms and make fluorescent silk. Other groups made a cellular laser based on GFP. Bob Burlage put GFP into bacteria in a way that would be activated and would appear if there was the explosive TNT, which he knew leaked from landmines. And so his idea was to spray a field. This is an example of one of the fields he sprayed. Spray a field with the bacteria. If there was landmines that would leak TNT, and then you could come back at night with a UV lamp and see where the fluorescence was. And that would show you where the landmines were. I know of at least 3 other groups that have worked on similar projects from starting with his initial work. But it's still a problem because you don't want to have any false negatives. You want if it says it's safe, you want it to be safe. And so people are still working on it. And GFP has appeared in art. The artist Eduardo Chach arranged to have Alba, a rabbit expressing GFP made by a French biotech company. And he would show Alba, who was his family pet, at art shows that he would have to get people talking about the interaction between science and art, or science, technology and art. And one of my favorite uses of GFP is in the Ang Lee movie Hulk, to explain why this man has the green color that he does. I actually know the screenwriter of this movie, and because his daughter and my daughter went to school together, and I asked him, how did he know about my work? And he said, I have no idea what you work on. And I said, well, you used it. It explains why the Hulk is green. And he said, oh, we had a student from MIT and he suggested that. So fine, I don't care how they use it. It was very amusing. Now, in the United States, there's a children's book. I don't know if one sees it in Malaysia or not. It's called If You Give a Mouse a Cookie. And in this book, this is a very troublesome mouse, because if you give the mouse a cookie, then he asks for a glass of milk. And if you give him a glass of milk, he asks for a straw. And if you give him a straw, he asks for a napkin. And it never stops. He keeps asking for more. I actually think this is a wonderful metaphor for scientists, because you give them a perfectly good green fluorescent protein, and the first thing they ask, do you have any other colors? Do you have any other things that you can show us? And so that's what scientists are. But the person who really did this was the third person to share the Nobel with us, which was Roger Chen, who made the first blue fluorescent protein, BFP, and then the first yellow-green, which he called yellow fluorescent protein. These were all done by modifying the sequence of GFP. But then, a couple years after that, Mikhail Matz and Sergei Lukyanov in Moscow, knowing about corals, realized that corals were also fluorescent organisms, and they had red fluorescence. And they isolated the first red fluorescent protein. And then many other experiments by Roger's group led to really a whole extensive spectrum of colors, ranging from blue all the way to deep red, and actually later even beyond that. And all these different colors allow one to do a lot of very interesting experiments. So I have to say I'm a little bit like this mouse wanting the other cookies as well. Because you can label nerve cells, as Josh Sain and Jeff Lichtman has done with Brainbow. You can look at exon activation, as Jeremy Nathans has done, looking in a female mouse and seeing having one X chromosome make a red fluorescent protein, and the other X chromosome making having green fluorescent protein. Since only one of the X's is working, the cells are either red or they're green. And of course, it can be used to find mutants, and we've done this. We have a nerve cell that normally in the larva, the arrow is pointing to where the nerve cell in cross-section is right next to the pink muscle. But as the animal becomes an adult, it gets completely surrounded by the skin, the blue. But there are mutants in which this surrounding never takes place. And as a result, the nerve cell remains near the muscle. And we can see this by putting RFP into the muscle, making it red, not pink, and GFP into the nerve cell. And you can see that in the normal wild type animal, there's a gap between the two. But in the mutant, where there hasn't been this surround by the skin, it's right next to it. And we can go and look for mutants, and we've been able to identify defects in at least 20 different genes that affect this process. So the question is, how do we make these discoveries? And if many or most discoveries are accidental, how do we increase our chance of making these discoveries? So what you do, as I already said, you do many experiments, hoping that at least something interesting will come up and that you are aware of it. You should do controls. We always say that people should do controls, but I'm going to say something a little different from this, and that is don't over control. And the reason I say this actually comes from a wonderful story by the wonderful scientist Seymour Benzer, who was talking about another wonderful scientist, Max Delbruck. And in the early days of bacterial phages, viruses that infect bacteria, you could do an experiment where everything was controlled. But they didn't always do that. And Seymour actually talks about it as the principle of limited sloppiness. You don't want to be too sloppy, but you also don't want to be too controlled. And the example he gives, or I've heard him talk about, is when they were first looking at the growth of the virus on bacteria. They made a lot of plates, and they stacked the plates up. And they'd leave them overnight for the virus to grow on the bacteria, come back in the morning. And they were a little bit lazy because they didn't actually turn off the lights. They just left the lights on and walked out of the room. And they found a very strange result. The top two plates always had more virus that came out of the bacteria than any of the other plates. This is a little surprising. They were stacking the plates just to save space. But when they finally realized what was going on, it turned out that the lights, the ultraviolet rays from the lights, were what were inducing the virus to grow and to come out of the bacteria. And it was only the top plates that were closest to the lights that were being affected. So this limited sloppiness, not putting all the plates at the same level on the table, but stacking them up, meant that they could make this rather unusual discovery. I think there's really two other things that I want to point out here. I think it's important to both question and wonder about things, to ask questions about all that's around us. And this means questioning things that are basically right in front of us that we've been ignoring maybe all of our lives. And not only question and ask about things that are around us, but also question our assumptions. So I'm going to give you some examples of this. The first comes from my own life. The very first scientific paper I was involved in actually had to do with the major transparent part of the body. Now, if I were doing this in person, I would ask everyone in the audience to raise their hands if they knew what the major transparent part of their body was. I can tell you that most groups, it's only about 5% of the people raise their hands. And then I give a hint. The hint is, it's right in front of your eyes. Because what I'm talking about is the cornea. But there is something that people have seen their entire life, have used their entire life. And we haven't, I had not thought about it until I went to work in this laboratory about why is it that this particular tissue is transparent? What makes it transparent? And people have studied that, but I think it's one of the big unanswered questions. Let me give you a couple more of these questions about things that are all in front of us and that we've been involved with our entire lives. So plants and gravity. An old experiment of plants is that when you grow a plant, the root goes down, the shoot goes up, you put a plant on its side, and the roots turn to go down and the shoot turns to go up. This is well known. But what isn't as well known is that this response to gravity doesn't only happen with the roots and the shoots. So what I'm about to show you is going to ruin your lives because you are never going to look at trees in the same way. Because some trees, called the set point of where the branch is main to the main tree. So some trees are like the cypress, and their branches are sticking straight up. Others come up at an angle. Others are completely horizontal. And some plants are, in a sense, mutant because they can't respond to gravity. And they are like the weeping willows, where the branches just hang down because they can't point up at any particular angle. And I'll give you a third example of something that is all around us, but I at least had not noticed it before. And this story comes from when I was a graduate student. I used to help teach the physiology course for medical students. And in that course, I asked, or there was a day, usually in October, in which a professor would come up and he'd start talking about the different types of muscle. Now, there's two different types of skeletal muscle. Now, we're not talking about smooth muscle or cardiac muscle, but skeletal muscle. One type is a muscle that works for a very short time, but quickly, and can go into oxygen debt. because it's only working for a small amount of time, it doesn't need to have a constant supply of oxygen. These are called the fast twitch muscles. Now, the other type of muscle doesn't work quickly, but continuously, usually for posture. And this continual use of the muscle means that it absolutely needs to have some sort of oxygen supply. And so it gets its supply of oxygen from a hemoglobin-like molecule in the muscle called myoglobin. And myoglobin, like hemoglobin, is red. It's dark. Now, I should tell you that at about this point in the lecture, most of the students were asleep. They had, oh, we're getting more information. We don't have to do it. And then the professor would say, so when you go home a couple of weeks for Thanksgiving and you're having turkey or you eat some chicken, you can now tell your parents why there is a difference between the white meat of the breast and the dark meat of the leg and thigh. It's because one is a fast twitch muscle and the other is a slow twitch muscle. If you're like me, you've seen chicken or turkey your entire lives. I don't know anybody who has really asked themselves the question, what is the basis between the two different types of meat in the chicken? But it turns out slow twitch and fast twitch. The legs and thighs are for posture, keeping the animal upright. The wings are supposed to be for flying, which they don't really do, so they don't work them that much. They don't need the oxygen supply. There's a puzzle that I used to enjoy when I was younger. I still enjoy them. They're called matchstick puzzles. And this gets us to talking about the idea of assumptions. Now, these puzzles are sort of fun. You're given a bunch of matchsticks. And the puzzle is to move the matchsticks, in this case, 3 matchsticks, so that you change this pattern to get 3 perfect squares. And you sit there and you move things around, you think about what it is, and maybe eventually you get to the point where you say, that's how I have to change. Those are the 3 matchsticks that I have to change. And look, I have 3 perfect squares. And you feel very proud of yourself for doing that. Another puzzle is this one of this fish. And for this fish, move 3 matches so that the fish turns around. And after a while, you might realize that you can move 3 matches, these 3, and now suddenly the fish is pointing in the other direction. So these are good. They exercise your brain. They make you think about how to solve problems. But then you usually get a really strange problem, and that's this one. Here are 6 matchsticks. Make 4 equilateral triangles. And you look at this and you try to move them around as you moved around the matchsticks for the other puzzles, and it's very hard until either you find the answer, or somebody tells you the answer, or you figure out that maybe your assumption was wrong. What is the assumption that is wrong? You've been thinking that it has to be a pattern in 2 dimensions. Maybe it doesn't have to be a pattern in 2 dimensions. Maybe it can be a pattern in 3 dimensions. And if it's a pattern in 3 dimensions, all of a sudden things fit. But this is not actually the only solution to this puzzle, because there's another assumption here. And that is that all the triangles should be the same size. It doesn't say they should be the same size. Why should they be? Maybe one could do this. Here you have 1 triangle and another triangle, 2 large ones, but then there's a small one and another small one right there. 4 triangles, and now it's more or less planar, not exactly. A completely different way of doing it. So 2 different assumptions. And then to make things even worse, one can have an even worse question. This one, make these 3 matches into 6. Now, you're not supposed to break the matches. That would be the easiest thing to do, is to just break them. But that's not the answer. The answer, again, is understanding about our assumptions. And the assumptions here are, why are we only thinking about shapes? Maybe it's Roman numerals, and the changing 3 to 6 just means moving it so that you have VI. So it's 6. And you might think, these are nice little puzzles, but what does this have to do with science? And a good friend of mine, former colleague of mine here at Columbia, is this man, Mu-Ming Pu. And Mu-Ming is having an illustrious career in neurobiology, just an astonishing scientist, making one discovery after the other. And I remember one day we were talking, and I can't remember who asked him, but they said, Mu-Ming, where do you get your ideas? How do you get all these ideas for these experiments? And he says, it's really easy. And subsequently, he called it the retrospective approach. He said, I look in a textbook. For him, it was the 2nd edition of the Molecular Biology of the Cell. He says, I read the textbook and I find an interesting finding. And I ask myself, how did they know that? And he usually realizes that they didn't know it, or there wasn't a lot of very good evidence for it. It's just what everyone believed. And so he would design the experiment that would actually test that hypothesis and either greatly add to it or actually show that it was incorrect. And he'd publish another very high-profile paper because he had made another discovery. And then he'd go back into the textbook and find something else that he would be interested in. So here's somebody who actually does constantly question the assumptions and ask, why do we know what we know? So I'm now at the end of my hour. I want to leave you with my favorite quote. One more thing here, then he publishes the correction. I want to leave you with my favorite quote about useless information or basic research. And this was a quote that was said by a different Robert Wilson, not Robert W. Wilson, but Robert R. Wilson. And Robert R. Wilson was a physicist, a sculptor, an architect, and the first head of the Fermi Lab in Illinois, which until the super collider in Europe came online, was the biggest particle accelerator in the world. Wonderful discoveries were made and are still being made at it. And in 1969, Robert Wilson was asked to come to the US Congress because they were thinking of funding the building of this big particle accelerator. And of course, it was 1969. That was in the middle of the war in Vietnam. And it meant that resources should be used for this science project. And so the people that invited Wilson to talk to the members of Congress wanted him to assure them that this was going to be something important because it would help national security. And so that was the first question they asked him. They said, Dr. Wilson, tell us, how many ways will this particle accelerator help the national security? And he said, none, which was not the answer they wanted to hear. So they asked him the question again. He said, no, it's not. That's not what this is for. And finally, John Pastore, the senator from Rhode Island, said to him, no, look, Dr. Wilson, in what respect will this particle accelerator help the national defense? And Wilson, he tried to answer the question so many times. Wilson said the following. He said, it has only to do with the respect with which we regard one another, the dignity of men, our love of culture. It has to do with whether we are good painters, good sculptors, great poets. I mean, all the things we really venerate in our country and are patriotic about. It has nothing to do directly with defending the country, except to make it worth defending. Thank you very much.
Thanks, Marty, for that crazy ending. That was really thought-provoking. So what was the outcome then? So is that
They built it.
they built it?
They built it, yes.
Yeah, Formula.
He
Yeah.
was able to convince them. I think they were going to build it anyway.
Yeah.
He actually, I was fortunate. I visited there, maybe about 8 years ago, and there's a main tower in the middle of this vast area that has the accelerator. And in this area, because he was the architect of it, there's this wonderful observation level of this tower that you can look out over everything and see the entire area. And the story was that when he was designing this building, he hired a helicopter, and he kept saying to the pilot to go higher and higher and higher until he found the right elevation where the view was the best he could imagine. And so he said, okay, how high are we now? And the guy told him, the pilot told him, and he said, that's how high we're going to build the building. He built the building for it. And all over the grounds are his sculptures that he made. Rather remarkable individual.
All right. Okay, that's interesting. I would
I'm
like
happy to take questions.
Yeah. I would like to kick off with the last one, the second last one from Muming, taking old hypotheses or unknown or unproven facts. Now, my question is a bit more grounded on realism. Funding. How does he find the funding to do something that is sort of accepted, sort of basic theory that everybody has gone over? How do we overcome that?
So this is a very interesting question. I hope I can say this quickly. One of the things that I have been very grateful for has been the fact that the way research is funded in the United States. I am funded by the National Institutes of Health, and I'm funded by a grant. Now, many people outside of science only know contracts, legal contracts. Yeah. And in a legal contract, what the situation is that you say, I promise to do A, B, C, D, whatever. And if I do all of those things, you will pay me. That's what a contract is. It's a bargain between people. But a grant proposal isn't that. A grant proposal is a promise that really says, at this point in time, these are the most important experiments that I can do. But, and we never write this in any grant, if GFP comes along or RNA interference or CRISPR-Cas9 or some other technique that, or some other finding that changes the direction of my research, I'm not going to waste my time talking about something I proposed 3 or 4 years ago. I'm going to do the more important experiment. And you're going to judge me, not did I do everything I said in my proposal, but what discoveries did I make? And so what we're really saying when we write a grant proposal is we are thinking about what the most important experiments are. And at that point in time, those are the most important experiments. We're not lying. But we're also saying we need to have flexibility. So when a new idea comes to us that we think is more important, we're going to use the money for that new idea. And if you think we made a mistake, don't fund us anymore. That's it. That's no good. But if we've made the right decision, and I'll give you another example, GFP. We could never write a grant proposal asking for funding to look at GFP as a marker. Because the experiment was you put it in and it either works or it doesn't work. So we didn't wait to get permission to do something. So when Mu Ming, I'm sure, writes a grant proposal, he doesn't say, I'm going to go look in a textbook and see if I can find something interesting. He's going to say, I've looked in a textbook, or I've looked in here, and I've realized that this fundamental idea that we have based so much of our thinking about how nerve cells work is not really well grounded. And so we're going to go and test that hypothesis. And this is how we're going to test the hypothesis using more modern means. So it's not that he's saying this is an important result. This is something that needs to be proven. And this is how we're going to go about doing it. So I don't think he ever had any problems any more than anyone else in getting funding, because he thought out what the experiment should be.
I agree. Fundamentally, I agree on that. I think I myself practice a bit of that, some flexibility into my grant. Yes, I do.
Wonderful.
Only thing waiting for that accident to happen.
We all do.
All right, there's one question from Miss Nur Naima. Thanks, Prof, for your great sharing. What advice?
Recording stop. Recording in progress.
What advice would you give to young scientists looking to make significant impact in their field?
So, this is I think that in general, what we all should be doing is the best experiments we can think of.
I agree.
At going after the problems, not thinking about how we network with people or who we're supposed to talk to or anything, but asking ourselves, is this an important question? Do I feel it's an important question to address? And that's going to be answered in different ways by different people. But for me, I think it is important to have some flexibility, to question, to wonder about things. I have a long list of experiments I'll probably never do because I keep thinking of another one that I'd like to do. So there's a little bit of a problem. You have to decide on what you want to do. But I'm going to say this in a very strange way. I don't know what the university system is like in Malaysia, but in the United States, you have a probationary period, 6, 7 years. And if you succeed, you get tenure. And I noticed something very interesting when over the years at Columbia University. Every person that worried, am I doing the right thing? Am I, should I be talking to this person? Should I be publishing in this journal, or should I be doing this or that? Every one of those people that worried about whether they were doing things correctly, they never got tenure. And all the people that got tenure basically had one thing in common. They just went after the science. They tried to answer the questions that they were interested in. And so this didn't always, I'm not saying this will always be the case, but I really think more than anything else, it's you go after the science, you go after what you're doing. And that's
dogged pursuit.
important.
Dogged pursuit.
Yeah.
Okay. That's interesting. To go after what you want. I suppose it comes back to the basic persona that you are, what you want to do, what is it, what interests you, what piques your curiosity, and
Yes.
that's about it. Yeah, just doggedly pursue that to the end. Okay, that's
Yes,
nice.
I think that this is, we have, at least I feel I have had all my academic life, this wonderful freedom to investigate things that I thought were important. It's been a real gift to be able to do that. It's been a real joy to be excited about things and to see how they follow through. So I feel we're very fortunate.
On that note, on that freedom note, how is it in the States? Do you still have the freedom to pursue useless knowledge?
So, the point about and it's a little facetious to call it useless knowledge.
Of course. No. I understand.
It's not useless, but I think that everyone has to decide for themselves what direction they want to take. There are some people that want very much to apply what is known and to a particular problem. And we need those people. That's a very important thing to do.
The applied section.
But there are others that feel that there is so much more to learn, so much more to understand, that getting that basic knowledge is going to be fundamental to changing things. And it depends on, there's not a right or wrong in these things. I think it really is that people have to enjoy what they're doing. There's a story I like to tell about my daughter. My daughter is now 30, but this is when she was 12. I was elected to the National Academy of Sciences, and they have a ceremony where you come down and it's a very boring ceremony because 100 people are one at a time asked to come on the stage, shake hands with the president of the academy, have a picture taken with the president of the academy, and then walk across the stage and sign your name in the book that they have. And it's nice for you when you're doing this, but imagine all the people in the audience. Those people have come there to watch one person and have to watch all these other people,
other 99,
walking through this thing. It's astonishingly boring. They do read one sentence about you when you're walking across the stage to sign the book. And I thought, oh, I really like it that my daughter is here, but she is just not, she's going to hate this. 12 years old, how can she stand it? So I make my way, after I've signed my book, I make my way down to where my wife and daughter are seated. And I say to my daughter, you know, I'm really appreciative that you're here. It's nice to share this with you, but I just want to apologize because you must be bored out of your mind. And my daughter looked at me. Frankly, she often looks at me in this way, like I am the stupidest person in the world. And she says to me, Dad, don't you understand about all of those people? I said, what about all those people? She had been listening to those sentences. She said, Dad, every one of those people were the first person in the world to do or see what they did. The very first person. I'm very impressed with that answer. That's a wonderful, and that is true. That is part of the joy of what we have. Also, the reason why we're so frustrated all the time, because nothing works every time that we do it, because we're trying to discover something that no one else has done before. So it's both the joy and the frustration of the work of having these things. But to me, I really like that joy. I like the joy of coming up with an idea. I like the joy of coming up with an idea even when it's completely wrong. I like the fact that I can think of something new. That is appealing to me. And I think there are these various joys that come out of this, and we do make some progress. Things happen, and that's good.
Okay. Marty, in a different manner, slightly off topic again. Personal freedom aside, dogged pursuit aside, I'm not sure what's going on in the States, but over here, I feel that we are being grant disbursement wise, we are being coerced into a different direction that does not allow much of this freedom that we're talking about. Do you have that?
So I think there are people that feel this. I've met them all over the world, that they feel that they are constrained. Part of it is we have to hope, let me say this slightly differently. Part is our obligation to educate people about these wonders, these things that arise. In the United States, there used to be a senator, a Democratic senator. You'd think he'd be okay, but this is many years ago. He, every year, would come up with a prize. He would call it the Golden Fleece. Now, to fleece someone is to cheat them.
Yeah.
And his idea, this was William Proxmire, his idea was that there were people that were cheating the government by wasting government money.
Okay.
And so every year he would pick a research project and say, this is getting government funding and it's just silly. And he would make fun of it until one year he made fun of something that turned out to be a really important diabetic treatment. And he had to apologize.
Okay.
Then several years, many years later, like 30 years later, another group of Congress people decided that they needed to have they wanted to start a prize, but this time it was for science that sounded initially silly, not important, something you wouldn't want to support, but then turned out to be amazingly important.
Okay.
And this was called not the Golden Fleece, but the Golden Goose.
Okay.
The goose that laid the golden egg.
Golden eggs, yeah.
And in this, one of the ones, I'm happy to say GFP was considered for one of these prizes. But the one that I really liked was an experiment that was done. The scientists were trying to raise a special breed or mutant of, I think it was a rat. And they were breeding the mothers and everything. They were trying to get this particular, these offspring, and they were very concerned that they would protect them. And so what they decided is they would take the babies away from the mothers and just make sure all the conditions were wonderful. But they found to their surprise that the babies didn't thrive at all. So they gave the babies back to the mothers, and then with the mothers, the babies did fine. And instead of doing their regular experiments, they asked themselves the question, why are the babies doing fine? And what they discovered was that the mother rats were constantly licking the babies. And so they did an experiment. They took the babies from the mothers. They didn't lick them, but they took a little swab and they brushed the babies. That touch, that physical contact turned out to be exceptionally important to allow them to develop as best they could.
Yeah, I
so
remember
they
this.
thrive from the touch. Now, you could say, okay, so that's wonderful. We know how to make happy rat babies. But it actually turns out that that same technique was then used in premature baby wards in hospitals all over the world.
Yeah.
The premature babies also needed to have that touch and that contact and that interaction.
Correct.
As a way of having them thrive as well. So immediate medical consequence. I think we should be telling more of these stories to say, we can't predict these things. We can't tell you where the next breakthrough is going to be. But there is, I think, a problem. I think that non-scientists have divided scientists into the ones that make that are interested in applying science.
Yeah.
And then the rest, and they use a very funny phrase. They say, oh, you're just curiosity driven.
Okay.
And what I think they mean by that is you don't care about us.
Yeah.
You don't care about anyone. You're only concerned about your own curiosity. But I can tell you, every scientist that I know and every grant that they've ever written has said, we are constantly thinking about the implications of our work, what it means more broadly.
To society, to the broadly, yeah.
Scientists are not just saying, oh, I'm going to do any experiment I want. We're going to say, there's a lot of interesting experiments we want to get to the bottom of, because we can tell you exactly why we think this is an important experiment to do. And I think we also have to convince funders of that idea as well, that we are, it's not ivory tower and I'm just doing whatever I want and I don't care about you. I care desperately about what the implications of my work are. So, and I think most scientists do too. Depends. Sometimes there's a lot of implications, sometimes there's not.
Yeah. I agree. We need some crusaders here. All right. There's a question from Professor Roslan. Prof. Chalfie, thanks for the wonderful talk. What is your advice to young scientists in a developing country like Malaysia who wish to do great science? Young scientists in a developing country like Malaysia.
So I've sort of already outlined a little of this.
Yep.
I think to do great science is asking questions, questioning assumptions, thinking about what the problems are and what you think is an important problem. I think what is often it feels like a problem is that people sometimes feel I have to have the latest equipment. I have to have all these different machines and technicians and large labs. And interestingly enough, I think most of the real discoveries are being made by people that sort of just go off and do something different and are doing it themselves. I think there's a lot, you do need some funding. I'm not saying you can do this in your basement or your kitchen, but I think that there are, I feel, questions all around us. And one question leads to another. And I think it is important, probably more, to think about what the questions are, and then come after the applications. If someone were to come and say to somebody, from one country to another, and say, I have made this observation, I have thought about these things, and now what I really need is your equipment, your techniques, or your ways of looking at this, then that sets up a collaboration. So this is not a good answer, because the real answer is, yes, there should be much more support of basic science, fundamental science all over the world. And we should all be working towards that. But I'm sort of a big one for collaboration and communication among people and interacting. And I think that's an important way of sort of circumventing whether one needs a piece of equipment or whatever. Not as pleasing an answer as I'd like to give, but at least one way maybe around this.
Okay. Yeah, that's definitely a point to look into. Educating the fund evaluators and increasing fund for more basic research, not so much looking into the applications. There's another question from Ms. Mas Jamila, or Dr. Mas Jamila, I'm not sure. Thank you for an eye-opening sharing session. My question is, in the process of finding new discoveries from useless information, I believe there could be unsuccessful points or experiences. Can you share them, and how do they, the scientists, move on? Thank you.
So I am not a good person to answer this question, and that has to do with the fact that when I was an undergraduate, I worked in a laboratory for the summer after my junior year. And it was the worst experience of my life. Nothing worked. It was absolutely horrible. And at the end of the summer, I said to myself, okay, you have proven to yourself, you are not a scientist. You cannot do science. Quit. And that's what I did. In my senior year in college, I took all sorts of interesting courses. I enjoyed them immensely. And when I graduated from college, I did various jobs. And it was absolutely the furthest from my mind that I was going to be a scientist. So how did I deal with being unsuccessful? I quit. And then after a couple of years, I got a job teaching high school. And after I made a discovery when I was teaching high school, and that discovery was that high school students get the summer off, but high school teachers have to find a job for the summer. And a friend of mine suggested that I go and work in a lab of someone that she knew. And I went to that lab, and that's where I did those experiments on the cornea. And to my amazement, I not only had an idea of an experiment to do, but I found somebody I could ask advice about how to do the experiment. He told me, and the experiment worked. I was astonished and somewhat seduced by the fact that I was able to actually do an experiment. And that brought me back into science. What I tell people now about how to deal with being unsuccessful is an answer that no one likes to hear, but I will say it anyway. When I started my postdoc, I started looking for mutants that were insensitive to touch, and it was easy to find these mutants. Now, animals can become insensitive to touch or any other sense in two general ways. One is development. The cells that develop to become the sensors don't develop. So your mutants are going to help you understand about development. The other type of mutants are those where the cells develop perfectly well, they just don't work. And they're going to tell you how the cells can actually do the sensing. So two very different problems: cell development, cell sensing, all coming from the same two pot of mutants. And I realized that this was a gift, because when one set of experiments were not working, the other set did work. So that led me to this idea that if you're doing only one thing and it's not working, you're not going to feel very good. If you're doing more than one thing not working, that's okay. And when I'm talking to graduate students, I usually say, have you ever noticed that an individual graduate student in a lab may be unhappy with his or her work because it's not working, but the head of the lab, who has a number of graduate students, at least somebody's work is working, and so they're happy. So it's this idea of doing, if something is not working, do twice as much or do another thing while you're waiting for the first thing to sort itself out, where you get another idea. So one way of coping with being unsuccessful is not to always be fighting just that battle, but to do other things as well. So that something is going well while you're waiting for the other to work itself out. You're muted.
Sorry, there's quite an answer there. But my personal take home on that is that summer school teachers are not paid. I didn't know this. Okay.
That's true.
That's crazy. Okay. But personally, I would like your opinion on this. Personally, I feel that a lot of times my personal curiosity is piqued when I teach students, whether it's in a laboratory setting or demonstration or even classroom setting, and I'm forced to explain things deeper and deeper, or crazy questions come out, and I'm said, oh shit, where do I get that answer for that? And I have to do a lot of homework, and I said, oh, there's a whole bunch of things waiting for me here.
I completely agree. It's one of the real joys of teaching and explaining and working with other people. A lot of the back and forth in my lab is actually diagramming things. How do we put this all together? How do we draw it out? And it's the discussion that gives rise to the ideas. It's not somebody just comes in and has the ideas out of the discussion and interaction.
Crazy. Okay. There's one from Dr. Sharifa. How does one measure the quality of science? What do you think of the H index that people use to measure their achievement in research and become obsessed with it?
So I never look at that, so I'm not a good person to talk about that. I get excited about the ideas. I want to, when we have people that come to apply for jobs, I am very interested in how they think about their work. If I get excited about how they are working, then I actually don't care what they're working on. I don't think they have to work on something that's going to be directly applicable to my work or not. I just want to see enthusiasm. I want to see involvement. I want to see an interesting hypothesis, something that I'd never thought of before is what I look for. I want to learn from other people. And that's what excites me about people that have come through, not where they publish or how many papers they've published, but how are they thinking about their science? I should tell you that in the United States now, there's a very big effort. I shouldn't say a very big effort, an effort to think about how we hire faculty. And there's a really remarkable man who's the chairman of the molecular biophysics and biochemistry department at Yale, named Enrique de la Cruz. And Enrique has been thinking a lot about this problem of how do we hire people? How do we hire people in a way that is fair to them, that we pick out them? And so, because the problem, frankly, here in the United States is you look and where did they go to college? Who did they work for? Where did they publish their papers? It biases everything that we look at. And so he has pioneered an approach. People in our astronomy department, I was talking to today, are doing the same thing, and we're thinking of doing this in biology, where the first phase of the application, now eventually you have to get letters of recommendation and everything, but the first phase of the application is write us a description of what work you've done and what work you want to do, but don't indicate anything about yourself in this. You can't say who you're working with, where you're doing the work, where you got your
publish,
where you've published it, whether you're a man or a woman, whether you're anything, nothing that indicates anything about you.
Nice.
Everything has to be out of the application. And what they do is they go through those applications saying, what about the science excited us that we would like this person as a colleague? And from that, they get a short list of people. I think they went from like 160 to about 30. And they were quite surprised at the variety and diversity of people whose science excited them. And it wasn't exactly the group of people that they would have picked out had they known, oh, I want to make sure we get this person from Berkeley and this person from Stanford and that person from Princeton. No, none of that. Who was doing interesting science? And I've heard now about these sort of where people are anonymous in the initial application. There's a man a scientist at UCSF, Keith Yamamoto. I think Keith is one of the most amazing scientists I've ever met, and one of the fairest people I've ever met in my life. And he said a remarkable thing the other day in a meeting that we were both in. He said that he was asked to be on a panel for the Gates Foundation. And in the Gates Foundation, all of their applications, they have to start off with, I think, a 1 or 2 page description of the work. Again, no indication of who it was that wrote the thing. And he said, and this is somebody I consider extremely fair. He said, the people I chose were not the people I would have chosen had I looked at where they came from or who they were or who they worked for or where they published. So that change, I think, is really important. I think that I hope this is going to happen more and more, that people will be chosen by the merits of their thoughts and their ideas. Who they happen to have worked with or where they happen to have been able to get their papers published or any of that should be the science. So I'm excited about this idea. This is a sort of a new thing right now. To me, I think it's a very exciting possibility.
This is the first that I've heard of it, and it sounds very interesting, very promising. I personally feel that attaching numbers to a person, H-index, a number of publications, it just shows you can do the measurement, but you can't.
About 10 years ago, I was in Europe, and I happened to be, I was on a trip or a couple of trips where I went to 2 different scientific institutes in 2 different countries. And the heads of both of them came and they were showing me around, and they both said exactly the same thing. They said, this year has been a great year for us. We have published more papers in high impact journals than we ever had before. And I said, okay, that's nice. What was discovered? And they didn't know. They couldn't say what had been found. What were the results that led to those papers? All they knew is the numbers. And I obviously think that that's a very bad thing.
Yes, that skews a lot of stuff. That's a question from Anwar Hashem. Before I read the question, the floor is open to anybody to ask a direct question to Martin. Okay. Prof, what is your insight forward about big data, data science, artificial intelligence, machine learning, especially precision medicine, into GFP, applied mass spectrometry, bioinformatics, etc., etc., etc.? Mouthful of words.
So that's a short question. I would say, I think we want to use every tool we can. And I think that anything that we can use that will help us analyze data, think about data, is important. I've done my own, I don't know how popular this is in your country, but one of the things that's in the news a lot here in the United States is all the concern about Chat GPT.
Oh, yes.
Or GPT-4.
Oh, yes. Oh, yes.
And the interesting thing about this to me was that the worries that we'd see people discuss on television was students are going to cheat. We are going to ask our students to write a paper about some subject, and they're going to go to Chat GPT or some other place, and they're going to write a paper, and we won't know if it's right or not because it's not their work. And it's this big worry that students are going to cheat. And I think this is ridiculous. And I did a test. So I was helped by a postdoc in my lab, and I asked for an essay to be written by ChatGPT, 1,000-word essay about a subject I think I know something about. Now, my name is I am the only Martin Chalfie in the world. And that's because my last name was something that came out of my grandfather when he moved from what's now Belarus to the United States. He changed his name, and so there are no other Chalfies. There certainly is no other Martin Chalfie other than descendants of my grandfather. So I decided that the essay that I would ask ChatGPT to write was a 1,000-word essay on the early childhood of Martin Chalfie. I wanted to see what it would find. What would it find? What would this essay be on? And it was more wonderful than I could ever imagine. It was wrong in everything. Now, because of the Nobel, there's a Wikipedia page about me. There is a biography on the Nobel Prize website that I had to write an autobiographical essay. So that information is there. But what this program did is it got my birthday wrong. It said that I had an older sister named Karen. I have 2 younger brothers. I don't have an older sister. It said both my parents were academics. They were not. They said my father was a high school biology teacher. My father did not graduate from high school. So it just was wrong in every imaginable way. And so I asked, we sent the essay back to ChatGPT and we said, are there any factual errors? And it said, no, there are no factual errors in this essay. So to me, this is the most wonderful thing a teacher could hear. I think instead of assigning a topic that the students should write an essay about, the teacher should give the topic to ChatGPT and say, write an essay on this topic. And then when they get the ChatGPT thing, which ChatGPT will not correct because it's perfect, you give that to the students and you say, correct this. And they have to go and actually do the research to figure out how to correct the essay. Okay. So to me, it's how you use these things, not what they are. If there's a way that machine learning can help us amass vast amounts of data and work with them in a way that we normally can't, I think that's fantastic. I think that big data can be very important. I know that doctors right now, talking to a doctor about a colonoscopy a little while ago, and he said, now we use artificial intelligence all the time in a colonoscopy. It's not that they rely on it. It's they're doing their observations, but there's a program running in the background that says, wait a minute, shouldn't you look at this point over here? Or shouldn't you look at this? Or shouldn't you look at that? And so it's extra. So it's adding to what the doctors can do. I can't see anything wrong with that. I think that's terrific as a way of increasing the abilities that people have. Anything that can help us broaden what we can do, I think, is good. I mean, that's what GFP is. That's what all the other techniques we've ever had is they enlarge our abilities. Why shouldn't these large data sets do the same thing?
Okay. All right. That's quite different from what I hear from one of my industry friends.
Well, I'm an optimist.
my industrial friend asked ChatGPT to write a Rust program, and he said the first version was with a lot of ChatGPT did come up with a complete Rust program, but had a lot of bugs in it. So he said he fed it back to Chat and asked Chat to correct it, and it says the second round it came out perfectly and it worked fantastic. So there are two different ways on how this could be used.
Why not? Yeah.
Yeah. Okay. Any verbal questions? Anybody? There's one on the chat. Hi, Prof. By Farid Sairi. Hi, Prof. Thanks for the great talk. In the current generation, information is not lacking, and you might have so many useless things that you are interested. How do you prioritize your curiosity?
Well, you know, I have to admit I'm sort of a random walk in some of these things. I have to get excited about if I think the idea is exciting and it will give me a possibility of understanding things that I'm working on. I will work in that direction. I think that this is probably more just myself, but I think that as one does work for several years, there are certain problems that you keep coming back to. There's certain things that you want to address, and you're always sort of looking for another way of addressing them. Is there a new way or a new approach or something? But the problems are the ones that have been sitting in the back of your head for quite a while. And so for me, the priorities are, they've been sort of the same for a while, but every once in a while there'll be a new approach and I'll be excited about that, at least for a moment, or at least until I can convince somebody in the lab that they should be interested in this as much as I am.
Okay. I suppose that's about here, Prof. I don't think we have any more questions, and I would like to have your last word on this, on how we go about our scientific lives. And after that, we'll open up for a picture session before part ways.
Right. There's I don't know how popular this is outside of the United States. There was a famous musical made into a movie too called Guys and Dolls. It was all about New York City and petty gamblers and petty crooks that were there. Very entertaining musical. But the musical was based on a series of short stories by an author named Damon Runyon. And in the Damon Runyon stories, the New York City stories about all these petty criminals, there was one criminal that every time somebody from outside of New York would meet him for the first time, they almost always said to him, "What do you do?" And he didn't want to admit, well, actually, I'm a crook. I'm a criminal. Never said that. He never wanted to say that.
Nobody would.
And so in the books, he always says the same thing. When somebody says, "What do you do?" he says, "The best I can." And I think that's a pretty good line for all.
That's a pretty good one. Yes.
You do the best you can.
Yes.
You try to contribute as much as you can, and
yes.
frankly, enjoy it.
Yep.
So with that, thank you all very much.
Thanks, Marty. That was fantastic. Before we part ways, Marty, just stay on a bit for a picture session. Please, everyone present in this Zoom room, please enable your cameras. So we'd like to have a cam shot of everybody. Thank you.
It's nice to see all of you.
Yes. Hey, Secretariat. Okay. Room 2, room 3. Oh, I didn't know Prof. Riza was here. Done? Okay. All right, we're done. Thanks, Martin. Have a
Great.
good night. Have a good day. Thanks for your wonderful talk. And not, it's more of education. It's more of an idea of how to progress in our science. Thank you very much.
Well, thank you all for listening. I appreciate it. And
I appreciate-
thank you again for
Thank
the
you. Good night.
invitation.
Yes.
Take
Thank you.
care, everybody.
Thank you. Thank you. Thank you. Bye. Bye.
Bye-bye.
Bye.
Bye-bye.
Okay, we wish everybody a good day. All right. Please don't forget to join us in a future series. There will be a QR code displayed soon for attendance. Till then, Eid Mubarak. Terima kasih and selamat berjumpa kembali.
Thank you. Take care.
Okay, thanks, Prof. Riza, for joining. You can use the QR code, guys.